I'd like to thank LabRoots for their invitation to present our work. I wanted to talk to you about new checkpoints and biomarkers. These are my disclosures. Now, immunology has offered hope for curing cancer for over 100 years, but up until the last 8 years, you couldn't go into your doctor's office and get a reliable anti-cancer immunotherapy. But that's changed. And what's changed now is that we have a new strategy. And that strategy is to stop the tumor from turning off the immune response. And that's been termed checkpoint blockade. The revolution in cancer immunotherapy has come from our deeper understanding of the second signals that regulate the outcome of T-cell receptor signaling. Now, specificity is provided by the recognition of the T-cell receptor of an antigenic peptide in association with MHC. A positive co-stimulatory signal is provided by the interaction of the B7 molecules, CD80 and 86, with the CD28 receptor on T cells. And this results in signaling which results in optimal T cell proliferation, cytokine production, and cytolytic activity. The surprise was that there are negative second signals. We identified the PD-L1 and PD-L2 molecules and showed they interacted with PD-1. PD-1 cytoplasmic domain became phosphorylated and recruits the phosphatase SHP2, which results in dephosphorylation of the proximal signaling molecules of the T-cell receptor and CD28 pathway. And this results in reduced T-cell proliferation, cytokine production, and cytolysis. We made this discovery about 20 years ago in a collaboration of 3 labs, my lab, Clive Wood's lab at the Genetics Institute, and Tasuko Hanjo's lab in Kyoto. And this identified the drug target to block the PD-L1, PD-1 pathway, and first showed that PD-L1 could be expressed in an ovarian tumor. Blockade of the PD-1, PD-L1 pathway can unleash an anti-tumor T-cell response. It results in enhanced activity of effector T cells, such as increased killing by cytolytic T cells and increased production of cytokines like gamma interferon. This has led to clinical translation, and there are now multiple PD-L1 and PD-1 antibody drugs. These have been tested in multiple clinical trials and are now FDA approved in 21 different types of tumors. This actually represents, it's estimated, about 43% of cancer patients are eligible for a PD-L1 or PD-1 immunotherapy. The PD-1 immunotherapies do not work everywhere. They don't work in glioblastoma, pancreatic, or colorectal, which don't have mismatch repair deficit. They can work from a high of 87% response rate in Hodgkin's lymphoma to a low approved rate of 13% in gastric, averaging about a 20 to 30% rate in most of the common solid tumors. There's clearly room for improvement. Now, the future is clearly combination therapies. PD-1 blockade is the foundation to build upon, and it can be combined with blockade of other immunoinhibitors, kinase inhibitors or chemotherapies, angiogenesis blockade, TGF-beta or HDAC inhibitors, PD-1 blockade plus immunostimulators, cancer vaccines, oncolytic viruses or CAR T cells, more novel agents such as probiotics or nutrients. In fact, there's over 300 different combination therapies which have been tested in mouse and found to enhance the response to PD-1 immunotherapy. and these are now being developed and tested in people. Clearly, not all of the combinations are going to work. The ones highlighted in red have been tested, worked, and are now FDA approved, and include PD-1 blockade plus CTLA-4, or PD-1 blockade plus cisplatins or nab-paclitaxel, or angiogenesis blockade. Despite good preclinical evidence, phase 3 trials of PD-1 blockade plus IDO MEK inhibitors or colony-stimulating factor receptor 1 did not do sufficiently well in phase 3 trials. So they're not all going to work. So where does checkpoint blockade function? Now, it's believed that CTLA-4 primarily works in the lymph node. This comes from the idea that the B7 molecules are primarily expressed on activated antigen-presenting cells, very rarely on a tumor, and that the interaction of CTLA-4 antibody blocks the interaction with B7. In contrast, PD-1 was initially thought to work primarily in the tumor because PD-L1 can be expressed on many tumors, also antigen-presenting cells, stromal cells, myeloid-derived suppressor cells. PD-1 can be expressed on the T cell. However, we also now realize that PD-L1 in the lymph node is also important and can be expressed on antigen-presenting cells in the lymph node. So PD-1 is important in both. Now, CTLA-4 blockade works by the antibody binding to CTLA-4 and preventing the interaction of CTLA-4 with B7. This removes the higher interaction, affinity interaction of CTLA-4 with the B7s, and it results in unopposed stimulation of B7s through CD28. Now, it's also been shown in mouse models by Alan Corman and others that CTLA-4 antibody can bind to and deplete T regulatory cells which express high levels of CTLA-4. However, this hasn't been shown despite considerable effort in people. Now, it's important to completely understand the function of the PD-1 pathway. And we asked, what was the function of PD-1 on regulatory T cells? And the take-home lesson of this section is going to be that PD-1 is an equal opportunity inhibitor of T cell function. Loss of PD-1 on T effector cells can lead to stronger T effector cells. But to our surprise, loss of PD-1 on Tregs actually makes more suppressive T regulatory cells. And this is the work of Catherine Tan and Juhi Kuchru in Arlene Sharp's lab, where we made the conditional PD-1 knockout. And this is recently published in the Journal of Experimental Medicine. Now, the approach was to selectively knock out PD-1 only on regulatory T cells. And to do this, we used FOXP3 in a floxed PD-1 allele. And this inducibly deletes PD-1 on Tregs in adult mice. Now, T regulatory cells can work by multiple interactions to suppress the activity of both dendritic cells and T effector cells. The Tregs can express high levels of CTLA-4 and bind and remove CD80 and CD86. The CD39 and CD73 molecules on Tregs can convert ATP into immunoinhibitory adenosine. The IL-2 receptor can compete for IL-2, and Tregs can make suppressive cytokines such as IL-35, granzyme B, IL-10, and TGF-beta. Now, when Catherine made Tregs depleted in PD-1. She tested them in an in vitro Treg suppression assay and found that Tregs lacking PD-1 are more suppressive than wild-type Tregs. Here in the black line is wild-type T effectors alone. And this is a cell tracker violet assay of T cell division. And you can see the wild-type effectors divide well. In blue is the wild-type effectors plus control wild-type Tregs that express a normal amount of PD-1, and they suppress. But if the Tregs are depleted of PD-1, as shown in red, the Tregs deplete even or suppress even better. And this was also seen in vivo. In NOD mice lacking PD-1 selectively on the Tregs, they're protected from type 1 diabetes. Normally, NOD, as shown in blue, wild-type NOD mice with normal amounts of PD-1 on the Tregs develop diabetes as they age. In contrast, if the NOD mice lack PD-1 on their Tregs, as shown in red, they don't develop diabetes as they age. They have a higher percent of FOXP3 cells in the PD-1 depleted. The percentage of proliferating CD8 T cells goes down in the PD-1 deficient mice, and the percentage of cytokine-producing CD8 T cells making gamma interferon and TNF goes down in the PD-1 deficient Treg population mice. Now, in another model of autoimmunity, EAE, inducible deletion of PD-1 and Tregs before disease induction again attenuates EAE. As shown in blue, normal mice with PD-1 on the Tregs develop EAE, but in contrast, if the Tregs are lacking in PD-1, they develop less severe EAE. Now, transcriptional analysis and analysis of phospho-AKT showed that PD-1 deficient Tregs have reduced PI3 kinase, AKT, mTOR signaling. The hallmarks of the PI3 kinase signaling pathway were less in PD-1 deficient Tregs, higher in wild-type Tregs. Similarly, the phospho-AKT, as shown in the right, in blue in wild-type T cells, you've got phospho-AKT, but in the PD-1 deficient Tregs, there was less phospho-AKT. Now, it's known and has been published by Krellin and others that increased PI3 pathway activity results in reduced Treg suppressive capacity. And we wondered whether this pathway was what was resulting in the increased Treg function. Now, PI3 kinase AKT signaling regulates glycolysis and mitochondrial functions. And here it's shown that PD-1 deficient Tregs are less glycolytic. Here in red, you can see they have a lower extracellular acidification rate in a seahorse assay, and the glycolytic capacity is less in the PD-1 deficient Tregs, and they have less glycolytic reserve. Similarly, the PD-1 deficient Tregs have an increased mitochondrial function. You can see here in red, they have an increased oxygen consumption rate, increased spare respiratory capacity, and increased maximal oxygen consumption rate, consistent with increased mitochondrial functions. Now, if this is the case, increasing phospho-AKT activity should reverse this. And here in the middle, you show the condition without an AKT agonist. And indeed, the PD-1 deficient Tregs are less suppressive, more suppressive. But if they're treated with an AKT agonist, the amount of suppressive capacity of wild type or PD-1 deficient Tregs is lost, and both suppress equally efficiently. So in summary of this section, what I'd say is that PD-1 restrains both T effector activities, as is well known, but to our somewhat surprise, PD-1 also restrains Treg activities. So PD-1 on Tregs limits their activation and suppressive capacity, and if you lose or block PD-1, you get better suppressive activity. Now, this coordinates very well with some recent papers from the Nishikawa lab in Nagoya. And they found that PD-1 blockade reinvigorates dysfunctional PD-1+CD8 T cells and enhances PD-1 Treg suppressive activity in tumors. Now, there have been a few reports of hyperprogressive disease, which occurs when PD-1 blockade can actually activate or expand tumor-infiltrating PD-1 Tregs. And this overwhelms the PD-1 effector T cells and actually results in a worse clinical outcome to PD-1 blockade. And the tumors of these rare hyperprogressive patients had highly proliferating FOXP3 Tregs, which contrasts with the reduction in non-hyperprogressive tumors. Now, the important thing for biomarker development is that the Nishikawa lab showed that the frequency of PD-1 positive CD8 T cells to PD-1 positive Tregs in the tumor microenvironment can predict the efficacy of PD-1 blockade therapies, providing a possible biomarker. What it also suggests is that depletion of these Tregs may help treat or prevent hyperprogressive disease. Now, CTLA-4 blockade is one of the best combinations with PD-1 blockade, but it's been somewhat of a mystery why it works particularly well. And this may give some reason for why. It's because CTLA-4 targets Tregs rather preferentially. So if PD-1 blockade makes stronger Tregs, the CTLA-4 can actually remove these more active Tregs, resulting in a particularly valuable combination. It also suggests that development of agents such as bispecific antibodies that target PD-1 on T effector cells but not Tregs might be even more effective than the standard PD-1 CTLA-4 antibodies. What are additional checkpoints that could be targeted for cancer immunotherapy? The B7 pathway has been a rich source of cancer immunotherapy targets, with PD-L1, PD-L2, CTLA-4 blockade developed. VISTA agents, ICOS agents are in development. B7H3 and H4 agents are in clinical trials. And we noticed that there was another B7 family member called HHLA2. It was known to have one receptor, TMI-GD2, with a stimulatory activity, but there were suggestions that there was also an inhibitory receptor. We looked at HHLA2 expression and other B7s in normal kidney versus clear cell renal carcinoma. And this analysis done by Sam Freeman compares in reddish normal kidney and in blue RCC. And what we saw very quickly was that HHLA2 was highly upregulated in RCC. PD-L1 is just a little upregulated, PD-L2 a little more, B7H3 upregulated. Surprisingly, B7H4 was downregulated, VISTA upregulated a little bit, ICOS a little change. There's lots of CD8 T cells in RCC. It's highly inflamed. So this said that HHLA2 might be a good target. And indeed, we found HHLA2 expressed in multiple tumor types, high in renal, pancreatic, adenocarcinoma of the lung and colorectal, high in AML, not in some other tumors. We made an immunohistochemistry antibody, as has Zhang, and we found that some RCC didn't express any HHLA2, but that some renal carcinoma tumors definitely expressed HHLA2 on the surface of the tumor cells. When we examined with Sabina Signoretti and Rupal Bhatt, HHLA2 and PD-L1 expression in RCC, what we found is that they weren't co-expressed. If they were co-expressed, and each dot here is one immunohistochemistry of one tumor, and what we see is we plotted a PD-L1 score or an HHLA2 score of each tumor. If they were co-expressed, they would be out here in the upper right quadrant. But in contrast, the PD-L1 scores are all along the left axis. There's not much shift to the right indicating HHLA2 expression. Similarly, the HHLA2, if they express highly, do not move up in PD-L1 score. So they're not co-expressed. In a rare tumor where we found that PD-L1 and HHLA2 were both expressed, they were expressed in different areas. This is sequential sections by immunohistochemistry. On the left is immunohistochemistry of HHLA2, and you can see it's expressed in this tissue down to the lower left. And in the next section, we stain for PD-L1. You can see it's expressed in this section up in the upper right. And you can see that the areas don't correspond to cross-expression. Now, we also found that HHLA2 is regulated very differently than PD-L1. Gamma interferon is well known to upregulate PD-L1. And here in this renal cell carcinoma line 786, PD-L1 indeed upregulates, interferon definitely upregulates PD-L1 expression. However, interferon doesn't upregulate HHLA2 expression, so they're differentially regulated. Now, the Chen and Zhang labs have shown that TMIG D2 is a receptor for HHLA2 and can result in AKT phosphorylation and can stimulate T cell activation. But there was also reports of an inhibitory interaction of HHLA2. So we did a screen with retrogenics using HHLA2 fusion protein and found that HHLA2 bound specifically to KIR3DL3, a killer inhibitory receptor. Now, the KIR family is composed of 15 different members, and we went back and individually examined the binding of HHLA2 to each of the 15 KIRs. And this is done by transfecting 293 T cells and incubating them with HHLA2 Fc fusion protein. If they bind, you get two dark spots. duplicates. And you can see that the HHLA2 Fc binds to KIR3DL3 and its known receptor, TMIGD2, but doesn't bind to any of the other KIR family members, or to PD-L1 or PD-1. And the affinity of HHLA2 for KIR3 and TMIGD2 is about similar. Now, the KIR3DL3 cytoplasmic domain has an ITIM motif, just one motif, consistent with an immunoinhibitory function. It has 3 Ig domains in its extracellular domain. It's also unusual among the KIR family in that it's what's called a framework KIR. Many of the KIRs are lost in different individuals, but the KIR3DL3 is always maintained and expressed. We made HHLA2 antibodies that block HHLA2 binding to KIR3DL3, and we also made 2 HHLA2 antibodies which are selectively blockers of KIR3 but not TMIGD2. We also made KIR3 antibodies that block HHLA2 binding to KIR3, and also antibodies that don't block. Now we then examined KIR3DL3 expression, which is pretty rare on T cells. We activated T cells with mitogen and looked at the expression of KIR3DL3. It's not expressed rapidly, but after a while, it's expressed on a moderate percentage of activated T cells. We then tested the effect of KIR3DL3 signaling in a Jurkat IL-2 reporter assay, making a Jurkat T cell expressing T cell receptor in KIR3DL3, and an artificial antigen presenting cell, a CHO cell transfected with a single chain FB anti-CD3 antibody and HHLA2. And this then drives the IL-2 promoter driving luciferase. And so it's a luciferase readout for T cell activation. And we showed that in this system, CD28 results in upregulation of T cell activation as expected. But if the antigen presenting cell expressed HHLA2, this activation by CD3 and CD28 was greatly reduced. So this shows that the KIR3DL3 is indeed an immunoinhibitory pathway. And this was published recently by Rupal Bhatt and other members of the lab and collaborators in cancer immunology research. Now, we then looked at the effect of blocking antibodies in this T cell activation antigen. And here in blue and red, we're looking at the fold T cell activation. So a rise shows that the antibody is blocking the pathway, resulting in enhanced T cell activation. And indeed, the 3 antibodies that block the interaction stimulate T cell activation. Here, the HHLA2 antibody that doesn't block the interaction has no effect. So the mechanism of action seems to be blockade. Similar to the KIR3 antibodies, the 2 that block the interaction enhanced T cell activation. The antibody that didn't block had no effect. Now, we then tested the effect of KIR3DL3 signaling in an NK T cell cytolysis assay. The NK cells we used are the NK92MI cells. These are NK92 cells transfected with IL-2, and they constitutively express high levels of KIR3DL3, in contrast to the parental NK92 cells, which don't express hardly any. So we found that KIR3DL3 engagement by HHLA2 inhibits NK cytolysis. Here you can see that a K562 cell is killed by the NK92MI cells, but that if it expresses HHLA2, the cell is killed less efficiently in each effector to target ratio. Similarly, another target cell, Raji cells with beta-2 microglobulin knocked out, show the same pattern. We also found that the KIR3DL3 and HHLA2 blocking antibodies enhanced NK lysis. Here you can see in the dark colors the control, and in the brighter colors the antibody blockade. And you can see that the blocking antibodies enhance the cytolysis. In blue, the non-blocking antibody does not. So our model here is that the HHLA2 can interact with TMIGD2, and in naive T cells have a stimulatory role, but that in certain tumors or antigen-presenting cells, the interaction of HHLA2 and KIR3 inhibits T cell activation, cytolysis, and cytokine production in a dominant way, and that blockade of this KIR3 HHLA2 pathway would enhance an anti-tumor immune response. So in summary, HHLA2 is a ligand for KIR3DL3, which is an inhibitory receptor expressed in activated T cells and NK T cells. Blockade of this pathway can enhance T and NK activity. HHLA2 is expressed in the majority of clear cell renal cell carcinomas, and HHLA2 expression is non-overlapping with PD-L1 expression. HHLA2 is highly expressed in multiple PD-L1 negative tumor types. So the HHLA2 pathway is expected to impact a large proportion of the PD-1 refractory patients potentially. Now, there are multiple challenges for developing agents in the HHLA2 KIR3 pathway. The KIR3DL3 expression is relatively rare, indicating that this must be a powerful or highly regulatory T cell for the immune response in order to have a pivotal role. The other challenge to translating this is that the HHLA2, KIR3, and TMIGD2 genes are not expressed in rodents. One member of the pathway was lost, then the others, and so you can't do a mouse model. You would have to add all 3 genes back to make a model. So you can't do it directly in rodents. It's expressed in subhuman primates and multiple other mammals, so those are possibilities. But there isn't an orthologous mouse model. So we're going to have to model this in humanized mouse or human xenograft tumor models in mice. And we're really exploring what are the best ways to model a purely human pathway in vivo models in mice or other species. And I'd be interested in your thoughts on that matter. So in closing, I wanted to basically talk briefly about what I think is the future of cancer therapy decisions. And I think this is a combination of personalized medicine and immunotherapy. We do want to sequence every tumor Every patient should be tested for tumor mutational burden because this will identify tumors that are likely to be more responsive. Indeed, mismatch repair deficient tumors such as MSI in colorectal, they can represent about 15% of colorectal, about 4% of total tumors. And these can have a 40 to 62 response rate to PD-1 immunotherapy. The other thing that sequencing will tell us is whether there's PD-L1 or PD-L2 genomic amplification. This occurs, as shown by Margaret Schippe and Philippe Armand, in virtually all Hodgkin's lymphoma. And those have an exceptionally high rate of response to PD-1 immunotherapy, 87%, because they're fundamentally addicted to the PD-L1, PD-L2 immunoinhibition. While common in Hodgkin's lymphoma, it does occur in 1 to 2% of other total tumors. Sequencing will also tell us whether the tumor is expressing viral antigens. And indeed, the immune response is most designed to respond to foreign antigens, such as papillomavirus, as can be found in Merkel cell lymphoma or head and neck. Sequencing tumors will also identify actionable mutations, which will allow the use of targeted kinases or cell cycle inhibitors. It'll also identify the neoantigens, which can be developed for cancer vaccines. This will let us choose and analyze the tumor microenvironment and profile it to look at PD-L1 expression, other mechanisms of immune evasion, such as IDO, TGF-beta, B7H4, cellular matrix, or other mechanisms of immunoinfiltrate, and also measure how much CD8 and other immune cells are infiltrating into the tumor. There's also great progress being made in vivo imaging of PD-L1 expression or CD8 immune infiltration into tumors. We're also rapidly being able to look at tumor DNA in the blood and tell a surprising amount about the whole tumor in the body and what's happening. This will allow us to choose the best immunotherapy and combine it with the best targeted therapy or vaccine. I think it's a really exciting time to be an oncologist or researcher because you can bring greater benefit to patients. The PD-1, PD-L1 blockade pathways work on a wide variety of tumors with a moderate percentage of responders and a good safety profile. It gives us a good foundation to build on. And with this success, really your human creativity has been unleashed. And you're going to get to do the experiments, which will really show us what works, what's safe, how it works, and who it will work for. These are the members of my lab who've done the work over the lab, including Julia Brown, Kwafan Kai, Yanping Xiao, Kathleen Mahoney, Saham Yu, Apurvi Chaudhry, Sarah Klein, Marika Speranza, Shang Bu, Ping Hua, Bao Guangzhu, and Yang Bao. Also, I'd like to acknowledge my many collaborators over the years, really always beginning with Arlene Sharp's lab in the investigation of B7 family members. The Treg work I showed today is a collaboration primarily done by Catherine Tan, Juhi Kutru, and Pete Sage. The HHLA2 work is primarily done by Abdullah Burgess and Kathleen Mahoney, with the strong collaboration of Rupal Bhatt's lab at the BIDMC with Julia Kong, Chung-Hua Chen, and David McDermott for RCC. And the PD-1, PD-L1 work really begins with the identification of the pathway with Clive Wood and Tasuko Hanjo. Thank you. I'd be happy to take further questions.